Welcome to the Dup Dive, where we plunge into the heart of complex topics to give you that shortcut to being truly well-informed. Today, we're taking on a microbe that is, frankly, a master of disguise: E. coli. For most of us, it's just a normal, harmless resident of the gut, quietly going about its business. But here's where it gets fascinating. Some strains acquire very specific genetic tools, and suddenly they transform into potent, even deadly, pathogens. Our mission for this deep dive is to unpack the different forms of E. coli infection, what the veterinary world calls colibacillosis, basically E. coli-caused disease, especially as it affects young piglets. We're going to explore how these tiny bacteria manage to cause so much trouble, the clear and distinct ways those troubles show up, and the innovative ways the swine industry is fighting back. You'll gain a quick understanding of a major challenge in animal health, with some truly surprising insights into what I like to call bacterial warfare.
And it's vital to understand the sheer scale of this challenge. These E. coli infections aren't just, a minor nuisance. They represent a major epidemiological and economic problem globally. Think about the impact. Substantial losses through piglet mortality, but also reduced growth rates, poor feed conversion, and the significant costs of medication. It's a pervasive issue that quietly chips away at the bottom line for producers worldwide.
So let's start with that surprising dichotomy. You mentioned E. coli as a master of disguise, and it's true. It exists as both a normal, harmless inhabitant of the pig gut and a significant cause of infectious disease. It's like having a Jekyll and Hyde character living inside, isn't it? What truly separates the good guys from the problematic ones.
You've really pinpointed the core distinction there. It's all in the genetics. The crucial difference lies in whether these E. coli strains acquire what we call virulence factor genes. These are like special blueprints for building tools that make them dangerous, and they're often carried on tiny circular pieces of DNA called plasmids. It's these acquired genetic tools that transform them from, harmless residents into potent pathogens.
That makes perfect sense. So these tiny genetic additions basically flip a switch from benign to devastating. And speaking of devastating, you mentioned the economic impact earlier. Can you give us a clearer picture of just how significant those losses are?
Absolutely. The numbers can be, quite stark. For instance, in affected litters, neonatal diarrhea caused by E. coli can lead to mortality rates as high as 70% if left untreated.
70%? Wow.
Yeah, and for edema disease, another form of E. coli infection, mortality among affected pigs can approach a staggering 90%.
90. That's almost everyone.
It's devastating. And even when it's not fatal, like with post-weaning diarrhea or PWD, the economic toll is still immense. While PWD might have lower direct mortality, it causes a significant fallback effect, meaning pigs grow slower, take longer to reach market weight.
Which drives up production costs.
Exactly, considerably. It's a hidden economic drain.
Wow, so the economic hit alone is enormous. But what truly struck me when I looked into this was how these pig infections, seemingly isolated, actually ripple out and impact human health and public health efforts globally. Can you tell us more about that connection? What are the broader implications beyond the farm?
That's an excellent point, and it highlights how interconnected animal and human health really are. While the specific E. coli strains causing disease in pigs are generally different from the ones that cause severe human illness, like the well-known E. coli 0157.H7.
Right, the hamburger bug people hear about.
Exactly. But pigs are still recognized as potential reservoirs for certain pathogenic E. coli strains. This raises a potential foodborne risk to humans through contamination of pork products during processing. Even more critically, the extensive use of antibiotics in swine production has contributed to the emergence of what we call multi-drug resistant, or MDR, E. coli strains.
Superbugs, essentially.
Basically, yes. Bacteria that have developed resistance to multiple types of antibiotics. And their presence in the food chain or environment is a significant public health concern for all of us, limiting our treatment options for human infections.
That puts a whole new spin on it. So, given these far-reaching impacts and the sheer cunning of these bacteria, how exactly does E. coli manage to cause disease in the first place? What are its core strategies that lead to such varied outcomes? Let's pull back the curtain on its cunning strategy, which turns out is a fascinating two-step process of molecular warfare.
That's right. The first crucial step is adhesion and colonization. The bacteria must first successfully attach to the cells lining the small intestine. They can't just float by.
They need to stick.
Precisely. They do this using tiny, hair-like appendages called fimbriae, or pili. Think of these Fimbriae-like types F4, F5, F6, F41, and F18 as specific keys
Okay.
that bind to equally specific locks or receptors on the surface of the host cells.
The lock and key analogy. Got it.
Yes. And this interaction is absolutely critical because it allows the bacteria to resist being flushed out by the normal movement of the intestine. Once they're securely attached, they can establish a colony and then proceed to deliver their toxins. It's a bit like anchoring themselves before launching an attack.
Makes sense. Anchor down first.
And it's also interesting to note that the locks for F18 Fimbriae aren't present in piglets until they're around 3 weeks old.
Okay.
Which is why we don't see F18 strains causing disease in very young, like newborn piglets.
Yeah.
The lock isn't there yet.
So they anchor themselves, they establish a foothold. And once they're securely in place, that's when the real trouble begins, right? Because the second step is toxin production. What's truly wild is how the specific type of toxin they produce fundamentally dictates the entire nature of the disease, leading to completely different clinical pictures.
Indeed. First, we have the ETEC enterotoxins, heat labile, or LT, and heat stable, ST and STB. These act locally, right there on the small intestine.
Okay, so staying in the gut.
Yes. Think of them as signal senders. They basically trick the host cells into opening up their internal water taps, causing a massive efflux, pouring out of water and electrolytes from the cells into the gut lumen.
Which leads to
That profuse secretory diarrhea. Crucially, these toxins cause a functional disturbance, a sudden rush of water, without physically damaging the intestinal lining itself.
So it's like messing with the plumbing, not wrecking the pipes.
Good analogy, yes. Now here's where it gets really interesting and fundamentally different. We then have the STEC toxin, Shigatoxin 2E, or CX2E, which is responsible for edema disease.
Okay, STX2E, different beast.
Totally different. Unlike the ETEC enterotoxins that stay local, CX2E is absorbed from the intestine into the bloodstream.
Whoa, okay, so it gets out.
It gets out and acts systemically, meaning it travels throughout the body. It's a potent cellular poison that specifically targets the cells lining small blood vessels, particularly in the brain, stomach, and colon.
Targeting blood vessels, that sounds bad.
It is. Once it binds and gets inside these endothelial cells, it shuts down their ability to make essential proteins, leading to cell death. This widespread damage to small arteries and arterioles causes the blood vessels to leak, leading to fluid accumulation in surrounding tissues. That's the edema.
The swelling.
Right, as well as internal bleeding and the formation of tiny blood clots, microthrombi.
That's such a critical divergence then. It's not just one kind of trouble, it's a shapeshifter. You've got ETEC, the local disruptor, causing purely enteric disease focused on the gut with that characteristic profuse diarrhea. But then there's STEC, the body-wide saboteur, causing a systemic toxemia that affects the entire body, leading to edema and neurological dysfunction, and importantly, often in the absence of diarrhea.
Exactly. That absence of diarrhea is a key clue.
It's like two completely different battle strategies in one organism, which is a truly critical insight for anyone trying to fight it because the signs you're looking for will be so different.
Absolutely fundamental for diagnosis.
Absolutely. So now that we understand these distinct molecular mechanisms, let's explore how they translate into the equally distinct clinical syndromes you might actually see on the farm. The specific Fimbriae and toxins combined with the pig's age truly create unique presentations.
Right. For neonatal colitis, it typically affects piglets less than a week old, usually between birth and 4 days of life, and the onset is very rapid.
Bang, it hits them.
Pretty much. You'll see profuse, watery, often projectile diarrhea.
Projectile, wow.
Yeah, it can be quite traumatic. The color ranges from whitish yellow to light brown. This quickly leads to severe dehydration, evident by sunken eyes and leathery skin, and without intervention, mortality can be very high. Litters from younger mothers, or gilts, are often more affected due to less developed maternal immunity passed on.
Okay, so that's the newborns. What's next?
Next, post-weaning diarrhea, or PWD. This syndrome typically strikes 5 to 14 days after weaning. This is a period of immense stress for piglets, involving abrupt dietary changes, mixing with new pigs, and a decline in their passive immunity from the mother.
A perfect storm, really.
It really is.
Yeah.
Here, the diarrhea is still watery to pasty, often yellow-white or gray-brown, but generally less severe than in neonates. While mortality is lower, a large number of pigs get sick. Affected pigs will show dehydration, loss of appetite, lethargy, poor growth rates, and a rough hair coat.
Contributing to those economic losses again?
Exactly. All contributing to those significant economic losses we discussed earlier.
Okay. Then the really weird one, edema disease.
Then there's edema disease. This one is particularly striking because it classically affects the fastest-growing, healthiest-looking pigs in a group.
The best ones, that seems cruel.
It does. Usually 1 to 2 weeks post-weaning. It's often peracute, meaning sudden death can be the first and only sign you observe.
Just find them dead.
Sometimes, yes. In pigs that are found alive, the clinical signs are predominantly neurological due to the brain swelling, the edema. You might see ataxia, which is a loss of balance, aimless wandering, stupor, and even lying on their side with paddling movements.
So brain-related signs.
Definitely. A highly characteristic sign is subcutaneous edema, which causes noticeable swelling of the eyelids and forehead. Looks puffy.
Ah, okay, you can see the edema there.
Yes. Sometimes you'll hear a peculiar high-pitched squeal from swelling in the larynx or voice box. Crucially, and this is a key differentiator we mentioned, diarrhea is notably absent in most cases of edema disease.
No diarrhea, but sudden death, neurological signs, swollen eyelids. Very different picture. That sudden death aspect, especially in the healthiest pigs, is incredibly sobering. And the neurological signs without diarrhea, it almost makes you do a double take, doesn't it? It truly flips the common perception of E. coli on its head.
It really does.
What about other forms? Is that it?
Well, finally, we have septicemic colibacillosis. This is typically seen in very young piglets, often those with inadequate protective antibodies from their mother's colostrum. Didn't get enough good colostrum.
So weak immunity.
Right. Signs include sudden death, lethargy, weakness, and fever as the infection spreads throughout the body via the bloodstream. Sepsis, essentially.
Okay. Given these varied and often dramatic clinical signs, accurate diagnosis is absolutely crucial. This involves a systematic approach that combines what you observe on the farm, what you see during a post-mortem examination, and then definitive laboratory tests. It's truly detective work, isn't it?
It absolutely is. On-farm observations and post-mortem examination give us critical initial clues. Clinically, the age of the affected pigs, the precise nature of the diarrhea or its absence, and the presence of edema or neurological signs are key for differentiation.
Putting the puzzle pieces together?
Exactly. During a necropsy or post-mortem for ETEC infections, the visible lesions are often subtle. You'll typically find a fluid-filled, gas-distended small intestine with yellowish, watery contents, but minimal obvious damage to the gut lining itself.
Okay.
For edema disease, however, the lesions are what we call pathognomonic, meaning they are uniquely characteristic and point almost definitively to that disease.
The smoking gun lesion.
Kind of, yeah. You'll see thick gelatinous swelling, or edema, within the wall of the stomach, particularly along the greater curvature, as well as in the mesentery of the spiral colon, the gallbladder, and in the tissues just under the skin of the eyelids, forehead, and larynx. That swelling is key.
So visible edema in specific places.
Right. For definitive laboratory confirmation, which is the gold standard, proper sample submission is vital. We need fresh intestinal contents, or fecal swabs, and tissue samples for microscopic examination collected quickly after death. Preservation matters.
Get them to the lab fast.
Yes. Bacterial culture allows for the isolation of E. coli, and the presence of beta-hemolysis on blood agar, meaning the bacteria break down red blood cells, is a strong indicator of a problematic strain, as most ETEC and edema disease E. coli strains are hemolytic.
Oh, okay, that's a helpful clue from the culture plate.
It is. Then histopathology, the microscopic examination of tissues, is crucial. In ETEC infections, you'll see large numbers of bacteria stuck to the villi without much inflammation.
Just hanging on.
Right. While in edema disease, the key microscopic lesion is damage to the small blood vessels vasculitis and the formation of tiny clots in target organs like the brain.
Seeing that blood vessel damage under the microscope.
Yes. And finally, genotyping, typically using a test called PCR, is the gold standard for confirming the exact type of E. coli. It detects the genes for specific fimbriae like F4, F5, F6, F18, F41, and toxins like LT, SDA, STB, and STECHIX2. Tells you exactly what virulence factors it has.
So you know precisely which keys and weapons it's carrying.
Precisely. And because resistance is so common, antimicrobial susceptibility testing is essential to figure out which antibiotics, if any, will actually work.
Right, no point using something it's resistant to.
Waste of time and money, and contributes to more resistance.
So what does all this intricate understanding of the bacteria and the disease mean for managing these challenges on the farm? It points to a really significant shift, doesn't it? A paradigm shift, away from relying primarily on antibiotics towards a much more comprehensive, integrated approach to colobacillosis control.
That's exactly right. In terms of immediate treatment, yes, antibiotics, whether oral or injectable, can be effective in the early stages of infection. Common examples include things like amoxicillin gentamicin or trimethoprim sulfamethoxazole. But critical supportive care, especially rehydration therapy with oral or intravenous fluids, is absolutely vital to manage that dehydration, particularly in the diarrheic forms.
Got to replace those lost fluids.
Crucial. For edema disease, specific antitoxin treatments may also be used to try and neutralize the Shiga toxins circulating in the pig system.
So treatment is one piece of the puzzle, but you mentioned a paradigm shift towards prevention. How did the industry shift gears, and what multifaceted strategies are at play here to stop the disease before it starts?
Prevention is truly multifaceted, and it starts with the basics. First, foundational management and biosecurity are paramount. Stringent hygiene is the cornerstone, because the fecal-oral transmission route means reducing the environmental load of pathogenic E. coli is critical. You have to minimize exposure.
Makes sense. Less bacteria around, less chance of infection.
Exactly. This involves rigorous all-in-all-out management of facilities, with thorough cleaning, disinfection, and complete drying between batches of pigs. Drying is key. Environmental control, like providing a warm, dry, and draft-free environment for young piglets, also significantly reduces their stress and susceptibility to disease. Keep them comfortable.
Minimize stress, maximize hygiene. Got it. What else?
Second, immunological control through vaccination is highly effective. For sows, vaccination before farrowing with specific fimbrial antigens, the keys stimulates passive immunity in their piglets.
Okay, so vaccinate the mom.
Yes, so the mother passes protective antibodies, specifically secretory IgA, through her colostrum, her first milk. These antibodies physically coat the bacterial fimbriae in the piglet's gut, blocking the keys from binding to the locks on the intestinal cells. Prevents attachment.
So the colostrum is packed with protection, but the piglets need to get it.
Absolutely. Ensuring piglets get adequate colostrum immediately after birth is absolutely essential for this to work. That first drink is critical.
Okay, so vaccines for passive immunity. What about the piglets directly?
Right. For piglets themselves, to develop their own active immunity against post-weaning diarrhea, oral live avirulin vaccines are used. These often contain the F4 and F18 fimbriae, and they help the piglet's immune system recognize the threat without causing illness, like a training exercise for the immune system.
Primes them for the real thing.
Exactly. For edema disease, injectable toxoid vaccines are used. These are genetically modified, harmless versions of the STS2E toxin. They stimulate the piglet to produce neutralizing antibodies that can inactivate the real toxin if it gets absorbed, preventing that widespread vascular damage.
Okay, so vaccines targeting both attachment and the toxin itself.
It's smart.
Very targeted. Third, nutritional management strategies are critical during that high-risk weaning period. Historically, pharmacological levels of zinc oxide were highly effective for PWD, but their use is now restricted in some regions due to environmental concerns about zinc and manure.
Right, the environmental angle. So what now? Other effective strategies include providing highly digestible feed and reducing crude protein levels. This minimizes undigested substrate reaching the hindgut, which pathogenic E. coli could otherwise thrive on. Less food for the bad guys.
Starve them out, basically.
In a way, yes. Acidifiers, such as organic acids added to feed or water, help lower gut pH, which aids digestion and directly inhibits E. coli growth. And emerging strategies include things like probiotics, prebiotics, and symbiotics.
The good bugs in their food.
Precisely. Introducing beneficial microorganisms or selectively feeding the good bacteria already there to improve overall gut health and resilience against pathogens.
Makes sense. Boost the home team.
Fourth, genetic selection for disease resistance is a powerful long-term strategy. This is really fascinating. It involves breeding pigs that naturally lack the intestinal locks for F4 and F18 fimbriae.
So remove the target.
Exactly, aiming to remove the lock entirely so the bacterial key has nowhere to bind. For example, by identifying specific genetic variations, like in the FUT1 gene, we can select for pigs with natural F18 resistance. It's breeding for immunity.
Wow, that's elegant, breeding the problem out.
It's a very promising long-term approach. Lastly, there's a growing field of alternatives to antibiotics. These include promising approaches like bacteriophages viruses that specifically target and kill bacteria.
Viruses killing bacteria. Cool.
Yeah. Bacteriocins, which are antimicrobial peptides produced by other bacteria. phytogenics derived from plant extracts with antimicrobial or gut health properties, and even passive immunity products like specific egg yolk antibodies, or IgY, which can provide immediate, targeted antibody protection in the gut.
Lots of innovation happening there.
Definitely, it's a very active area of research. Ultimately, the core takeaway is this. A thorough understanding of these distinct pathogenic mechanisms, E-tech, causing local hypersecretion in the gut, versus S-tech, causing systemic vascular damage, is absolutely fundamental. It's key for accurate diagnosis and the implementation of truly effective targeted control measures. You have to know your enemy.
And it strongly reiterates that the future of controlling E. coli in swine is a sophisticated, integrated approach that moves far beyond just, reaching for the antibiotic jug.
Absolutely.
It's about the synergistic application of these diverse strategies, from meticulous hygiene and targeted vaccination to advanced nutritional management and genetic selection. We're getting incredibly precise in how we fight these infections, understanding the enemy at a molecular level.
That's the goal. Precision medicine for pigs, in a way.
So, considering the incredible precision with which these bacteria operate with their specific keys and locks, and their targeted toxins causing such different effects, what might this suggest about our future strategies for managing other widespread microbial challenges? Not just in agriculture, but perhaps even in human health. Could we move towards approaches that primarily focus on blocking specific adhesion points, like jamming the lock or neutralizing specific toxins, rather than the broad-spectrum killing that antibiotics perform, which often has collateral damage? How much more could we achieve if we understood other pathogens with this same incredible molecular clarity? Makes you think, doesn't it?